Have you ever wondered if plants get diseases just like us? Well, maybe not a cold or a fever, but they definitely do get cancer. In our body, cells continuously divide for growth and repair. And naturally, there is a whole system of checks and balances to make sure that they only divide at a proper rate, not too much and not too less. But when these checkpoints are damaged, cells are left uncontrolled and they divide rapidly at an exponential rate, leading to the formation of a huge number of cells forming clusters called tumors, which ultimately lead to cancer. However, in plants, cancer is actually caused by a bacteria called Agrobacterium tumefaciens. What gives this bacteria the special property is actually a structure present in its cell. Just like any other bacterial cell, it has a not so well defined nuclear region called the nucleoid, which contains all of its genetic material or DNA. But it also has an additional piece of circular DNA called a plasmid, which is independent of the control of the nucleoid and just kind of freely floats around the cell. It is this DNA which can code for tumor-inducing properties. When the bacteria infects a plant, the plasmid enters the plant cell and forcefully joins itself with the plant DNA, where it gets transcribed, that is, the code is read out, along with the plant's own DNA. This makes the plant cells divide abnormally and start forming tumors, finally culminating in cancer. This cancer in plants is also known as crown gall disease. But how is this exactly useful to us? Well, actually, this small, simple and primitive organism helps us in one of the most cutting-edge technologies that exist in biology today: genetic engineering or gene editing. Take a simple scenario. Insects like beetles and moths are plant pests, which chew on the leaves and stems and finally end up damaging the plant. To reduce the use of harmful pesticides, farmers have adopted a different way. They mixed spores of a bacterium called Bacillus thuringiensis with water and sprayed the plants with it. The bacterium, which then ends up growing on the plant, produces inactive but toxic proteins which enter the insect when it eats the leaf. In the alkaline environment of the insect's gut, the toxin suddenly becomes active and kills the insect. Now, what if there was a way to make plants themselves produce these toxins on their own without the help of bacteria? And that is where we edit their genes. The gene coding for making the protein is first taken from the bacteria. Now, we somehow need a way to integrate this into the plant's DNA. But this is not that easy because you can't just inject the foreign DNA into the plant cell. It will just get digested in the cell like any other foreign body. For it to get actually read out, it has to be joined with the plant's DNA. And that is where our simple-celled genetic engineer comes into use. Remember that it already has a plasmid which can integrate itself with the plant's DNA? Well, we just take out that plasmid and cut out the tumor-inducing portion from it, join in or ligate the protein coding gene, all of these done using specific enzymes. The now recombinant plasmid is put back into the bacteria with techniques like heat shocks. Then we let this harmless but useful bacteria infect the plant naturally. The plasmid, now containing the pest resistance DNA instead of the tumor-inducing DNA, as usual integrates itself with the plant's DNA. The plant transcribes it too and starts producing these proteins. Now the insects eating this plant will get killed because these proteins are being continuously produced by the plant itself. And we have produced a naturally pest-resistant plant generation. This simple, primitive organism has actually saved us a lot of trouble that we would have faced to make today's genetically modified crops. Not only plants, but we humans do have extra chromosomal circular DNA too. How did it get there and what exactly does it do? Watch this video to find out.